All right, welcome everyone to the fourth week of the Early Career Scientist Symposium at the University of Michigan. We have had two great sessions with early career scientists the past 2 weeks, and we are now in our third and final session with the early career scientists themselves. My name is Teresa Pegan, and I'll be moderating this week. I'm a PhD candidate in EEB here at Michigan in Ben Winger's lab, and I study birds including using museum samples. So I'm really excited to be with you today at this event. So a few reminders. I guess first of all, don't forget next week we have our final keynote presentation with Pam Saltis. So that'll be the final event of the symposium and should be a great talk. For today, we're going to have two talks. First, a talk from Eric and then Laurel. And after each of those talks, we'll have about 5 minutes for audience questions specifically about the talk. And then after both talks, we'll have the panel discussion where we'll try to focus on questions that are kind of bigger picture and theme. And I just want to remind everyone two things about the Zoom webinar. First of all, if you have a question for the panelists or the speakers, please put that in the Q&A area of the webinar rather than in the chat. You can use the chat, but if you put a question there, we might miss it. So the Q&A is definitely the better place to put that, your questions. And secondly, if you'd like a live transcript from Zoom of the talks, you can turn that on in the lower toolbar on Zoom. So I'm going to go ahead and introduce our first speaker, Eric LoPresti. I'm just going to pull up my notes here. Eric LoPresti has been interested in plants and insects for as long as he can remember, an interest encouraged by natural history mentors in high school and college. After doing research in college and various ecology and conservation field jobs, he joined the lab of Rick Carbon at UC Davis for his dissertation, which focused on relationships of sticky plants and insects. Hoping to build off this field research on species interactions, he joined Marjorie Weber's lab at Michigan State University for a postdoc, and Eric is now an assistant professor at Oklahoma State.
Awesome. Thanks for that introduction, and thanks everyone for organizing this. It's been a year-long procedure. Let me see. Okay, can you guys all see my screen now?
Yeah, I think it looks good.
Awesome. Okay, so today I'm going to talk a bit about stuff that I've been working on for now, almost a decade. And before I delve into the nitty gritty, I just want to sort of give you a background on where I'm coming from and sort of why I study this about plants. So plants are really the foundation of most terrestrial food webs. And they go through a bunch of very disparate parts of their life cycle. So most of them start their life cycle in a dormant state, trying not to get eaten, crushed, debraded, rotten, desiccated, or even buried too deeply. And then potentially after a few days, potentially after decades, if the conditions are right, the plant begins growth for the first time in its life. And when those leaves pop up from the ground, they might be in a barren sand dune like this plant. It could be in a lush tropical forest living as an epiphyte on top of another plant. And each, the environment that they come up in presents new challenges: how to avoid getting fungal and viral diseases, how to avoid getting eaten, how to, compete for light, etc. And then at a certain point in its life cycle, maybe once in its entire life cycle in just a few weeks, or potentially decades later, and for many years, it enters a reproductive state. And that brings a whole new batch of challenges: releasing pollen, into the wind, drawing in effective pollinators, repelling ineffective pollinators, maturing seeds without them being eaten. And so at each stage in the plant's life cycle, a variety of traits dictate its success. And I'm biased on this, but I think the coolest plant trait is stickiness. I'm not going to try to convince you of that. But what I want you to take from this talk is that there are a lot of sticky plants. It's a really functionally important trait, in addition to being really common. It's a great trait to work with. And then it allows sort of this field ecology component to be linked with an evolutionary component. And one of the ways we do that is using herbaria. And so just to give you a brief background on stickiness, all parts of a plant can be sticky. There are plants with sticky flowers, with sticky seeds, with sticky fruit, with sticky leaves, stems, roots, bracts, calyces, et cetera, et cetera, et cetera. And this is often a really characteristic part of the plant, which leads to some really wonderful common names. It also leads to some very illustrative species names. If you find something that has the specific epithet viscosissima, you can be pretty sure that it's going to be really sticky. And plants are sticky in quite a number of ways. I'm not going to go into them all. The most common way is that there are little secretory trichomes on the plant, and these are called glandular trichomes, and they can secrete all sorts of substances, many of which are not sticky, but many of which are sticky. And so the exact compounds that are sticky are not known in most species. However, in some well-studied groups like the Solanaceae, tobaccos, tomatoes, it's a combination of acyl sugars. And then in other species like monkey flowers, it turns out to be mucilages and several other classes of compounds. And still other plants can be what I call physically sticky. And so they can have spines or trichomes that either trap insects or trap substrate, but don't have any sort of sticky compounds. And this is really common in the Loasaceae, the stickleaf family, as well as gallium, which you guys probably know is a weed around Michigan. And backing up a little bit, so sticky plants, like other plants, sort of form the foundation for these small food webs. There are fungal and viral pathogens. There are a number of herbivores that eat every plant, and then there's a number of predators that feed on those herbivores. And the nice thing about sticky plants is that these food webs are much simpler, and that's because most small arthropods can't actually survive on the surfaces of sticky plants. They end up like that unfortunate fly in the upper right. And so I'm going to talk to you about functions today in this short time, just about stickiness of seeds and stickiness of plant surfaces. But before I do, I want you to remember a few things. The first is that plants generally have one type of sticky tissue. So the plants that have sticky seeds are not necessarily the same ones that have sticky flowers or sticky leaves. Secondly, stickiness has a whole bunch of functions. I'm going to talk about just a couple today. And I don't, from these sort of functional ecological investigations, I don't think we can draw any broad evolutionary conclusions. However, for that, I would need to go out and study all sorts of plants in the field, which is obviously logistically impossible. And therefore, I think there's the way we can draw evolutionary inference is by, looking at collections of plants, largely in herbaria. The first type of stickiness I'm going to talk about is seed mucilage. And you guys probably are not all that familiar with seed mucilage, though you might have met it at some point. And so the two most common species you'll know this from are chia, which gives sort of any food that has chia in it, it's kind of slimy texture, and flax as well, which is why it's used in many breakfast cereals and granolas, because this mucilage makes a really nice binder. But basically, seed mucilage is a layer of dried pectins that are deposited on the outside of the seed coat. And when the seed is wetted, it forms into this really gloopy, viscid mass that you can see there stuck on the end of a forceps. And this is a really common trait. I'll talk a little bit at the end about how many species have it, but my current estimate is that probably at least 5,000 species of plants have it. And that includes a lot of plants you're familiar with. A lot of the herbs you eat that are in the mint or aster families have it. Arabidopsis has it, which is wonderful because we know a lot about the chemistry, the development, and the genetic control of it in Arabidopsis and other mustards. Other plants that have it are the plantains. Metamucil is actually just the husks of a certain species of plantain, and then a whole lot of plants. And I want to specifically ask, how does a mucilage layer protect seeds in different environments? And so about 40 years ago, Patricia Fuller and Mark Hay were wandering around the Mojave Desert, and they noticed that underneath wild chia plants, that there were these little balls of sand. And inside each ball was a seed. And what had happened is that the chia had dehisced its seeds. They had fallen onto the sand below the seed, or sorry, below the plant, and then gone through a wetting and drying cycle. And so the mucilage blew up, and then as it dried, it bound the sand grains to the surface of the seed, making these seed balls. And this is a really rapid process. You can see that those basal seeds basically went from fully imbibed mucilage to these little balls of sand in just about a half hour. And they suspected that this might be defensive against both vertebrates and invertebrates. And specifically in the Mojave Desert, the two biggest granivores are kangaroo rats and harvester ants. And they did an elegant little experiment to show that this was an effective defense against both species. However, I told you a minute ago that there are probably 5,000 species that have seed mucilage. So I wondered whether this was broadly defensive. And so I did a very similar experiment testing sand-covered seeds versus control seeds of a whole bunch of plant species across both monocots and dicots against harvester ants, since harvester ants are a really nice, easy to work with granivore that really do eat a lot of seeds and are ecologically important in many ecosystems across the world. And so just to show you what this looked like when I did it, so I put out these bare seeds and these sand-covered seeds, and the ants really struggled with the sand-covered seeds. So this ant's been trying for 7 minutes already, and it took about 20 minutes to finish this and to get enough sand off the seed so it could bring it back to its nest and consume it. And compare that to the ant that walked by with a bare flaxseed, which can just pick it straight up and bring it back to its nest. It's not surprising that this sand coating led to decreased granivory in almost all the species tested. However, there are two possible mechanisms on how this could work. The first is really obvious. There's this physical barrier, and I think that video probably put that, visually a lot better than I could ever show you with data. But you could also imagine that the best possible camouflage for any seed or any organism is to wear the substrate that happens to be right below it. And so, it's possible that there are both of these mechanisms going on. So we did another experiment where we manipulated the background color independently of the color of sand that was coating seeds. And perhaps unsurprisingly for anyone who studies ants, ants are not particularly visual predators, so this did not function as a camouflage. However, it would be really interesting to repeat this with other important granivores like birds, which are obviously much more visual. So let's say that seed, instead of falling into a sandy area with loose substrates, let's say it falls onto an area with hard packed substrates or on a rock like that basil seed in the upper right. Well, what happens after it undergoes a wetting and drying cycle? The seed binds itself really tightly to the ground. And to look at the effect of that in reducing granivory, we did the same experiment with the same species. And it was quite a struggle for the ants to extract those seeds as well. So what you can see that this ant is doing is actually a really cool behavior where it's sticking its mandibles underneath the seed and clipping the mucilage strands. And when it gets enough of them clipped, it can actually pry the seed off and bring it back to its nest for its nest mates to consume. And perhaps unsurprisingly, they also survive significantly longer, most of the species, when they were bound to the ground. And so this sort of, we showed that in the field, the sticking to the ground matters. We were sort of interested because when you looked at these, some of these were really tightly bound to the ground and some of them were not. Marshall McMunn and Vincent Pan, who were at Davis, and I were interested in whether, the amount of force that seed actually stuck to the ground mattered. And so Marshall developed a little assay to get at dislodgement force. And so here you can see that what Marshall is doing is measuring the force that it takes, the shear force that it takes to break those mucilage, the mucilage attachment to the Petri dish in this case. And I should note that this actually took a lot of force. And in some cases, it took more force than the seed sort of had structural integrity. So the seed was more tightly bound to the ground than it was, or more force was required to dislodge the seed than was required to break the seed. So these were really tightly bound to the ground. And perhaps unsurprisingly, the seeds that had a greater attachment force were removed less by ants. And so there was a lower removal rate of those with a bigger attachment force. And I should note that 3 newtons is like 10,000 times the mass of one of these harvester ants. And now in my lab, Vincent, who was an undergrad assistant at Davis and is now a tech, and I are looking at this attachment force and how it keeps seeds from washing away on various substrates, including on these eroding stream banks in these really intense spring rainstorms we've been having in Oklahoma. And then there's a bunch more mucilage stuff coming soon. This is sort of becoming a focus of my lab here at Oklahoma State. And one of the things that I'm working on doing, and which I started as a postdoc, is to assemble a mucilaginous seed list from the literature, the taxonomic literature, from the USDA germplasm repository, as well as from herbaria. And it's really nice that mucilage in most groups is well preserved in herbaria, and actually you can measure the force of this ecologically important assay from seeds that you pull off the sheets. And then ultimately, once we gather a whole lot more data than we have currently, the goal is to ask these big comparative questions about the evolutionary drivers of mucilage, which we think are somewhat habitat and climactic. Okay, so we've talked about sticky seeds. What about sticky plants? The plant surfaces, stems and leaves. Well, as you can imagine, if you have a sticky plant and you put it in a sand dune or other area of loose substrate, you end up simply with a sandy plant. And this has a botanical term, samophory, which means sand carrying in Greek. And lots of people have noticed this over the years. So William Beale, who is a professor at MSU and for whom the Botanic Garden and the Herbarium is named, he noticed this in plants in the upper Midwest, and he hypothesized that this sand entrapment probably reduced herbivory. Norbert Jurgens was working on plants in the Namibian desert, and he was the one who coined the term samophory, and he hypothesized the very same hypothesis. Simcha Levi-Dune was looking at sticky plants in the Israeli desert, and he made the same hypothesis. And finally, Ted Farmer was looking at plants in British sand dunes on the coast, and he came up with the same hypothesis. And so Rick and I decided, well, this has been sort of hypothesized for 120 years, and no one's tested it. And fortunately, California has a whole bunch of good sticky plants. And so we chose 2 to test this on. The first, Bronefolia latifolia, is this really big, wonderful plant in coastal dunes, and it's really sticky. It gets covered in sand and it has really tough leaves. So we were able to, with a damp sponge, just reduce the amount of sand, sort of every week, remove all the sand on some plants, stems and leaves. And then Navarisha melita is this little phlox relative that grows largely on sort of eroding slopes and nowadays on roadsides. And it's always covered in dust and sand and stuff. We found a population that was growing in this sort of hard-packed clay pan and was very sticky, but didn't have any entrapped sand. And so we added sand to half of those plants. And sure enough, when you remove sand, herbivory goes up on the sand verbena. And then on Navarisha, if you added sand, it cut herbivory by an awful lot. And this probably comes as no surprise to anyone who's tried to eat anything at the beach. Eating sand is not particularly pleasant. And so we had the same question as we did with the seeds, where, is this camouflage or is this sand just serving as a physical barrier? So we did a very similar experiment, a sand removal treatment, matching the dune sand treatment, and then very much non-matching the dune sand treatment. And sure enough, it turned out again that herbivory was far higher on the bare leaves, and no matter whether the sand matched the background or didn't match the background, we got lower herbivory. So how about a sticky plant that's growing in an area that doesn't have windblown substrate? Well, you don't really recognize how many small flies and wasps there really are until you start looking at sticky plants. And then sometimes there'll be hundreds, sometimes there'll be thousands of dead insects on these plants. And if you look a little bit closer, you often find scenes like this. There's a living insect. This is a stilt bug, which is known as a predator. It's actually really useful in biocontrol in tobacco and tomatoes. And what it's feeding on is a dead acylid or robber fly on this Metzelia. And once you've seen that a few times, you sort of wonder, well, is the presence of these predators, which are largely acting as scavengers on these plants, is this helping the plant? You know, by having these dead insects on the plant surface, is the plant attracting these predators, which, you know, are eating the dead insects, but certainly could be eating any herbivores as well, and therefore increasing plant fitness as, you know, part of a sort of classic protective mutualism. And so we tested this on a really nice columbine in coastal California, which has a really simple predator community. Is about 7 species of spiders and true bugs. And it also has 2 caterpillar species that eat the buds, flowers, and fruit. So they impose a really big fitness cost on the columbine every year. And most importantly, it's amazingly sticky. A plant, you know, one of these columbines, which is pretty spindly and doesn't have all that much surface area, might have 2, 3, or 400 dead insects on it midsummer. And so we did a very simple experiment again. Every week, we just removed all the dead insects from half of our plants. And if you remove that food source, sure enough, those plants have fewer predators, and the plants with less carrion and fewer predators, sure enough, have more herbivores, and they set less seed. So we would call this, you know, a protective mutualism. The plants are providing the insects with dead insects, food, and those predators are providing the plant with some protection. And we've done a number of sort of very ecologically focused investigations in these systems. And we, as well as a couple other groups, including one in Brazil, have shown that this is the same system is operating in quite a number of sticky plants. In fact, basically every sticky plant that this has been tested in has found to be beneficial. And one of the really cool things is that when you go into an herbarium and you look at it, you not only see the dead insects caught on the surface, but occasionally you see these predators, which presumably was living when this plant was pressed, you know, 50 years ago. And so we can really reconstruct a bit of the ecology just from, you know, that specimen on a sheet. And so I think that, you know, the majority of the talk hopefully convinced you that the stickiness has ecological functions. And the next question is really, how do we make informative evolutionary inferences? I can't go out and test, you know, each of these things on, you know, across the world on all these sticky plants. And so this was the focus of my postdoc, and I'm going to warn you that I don't actually have a whole lot of data on this. The projects have been turned into a lot bigger projects than I thought they were initially. If you read the literature, there aren't that many sticky plants. So, you know, I can assemble a list of sticky plants in a couple of months, and that has not been the case. And so the way that we've sort of gotten some data that's analyzable at this point is to get accurate data across clades. And so with Aaron Tripp at UC Boulder, who's an expert in the Acanthaceae, especially the genus Ruellia, we've looked at whether stickiness is evolutionarily correlated with pollinators. And we have good hypotheses to think that sticky plants should either be autogamous or potentially rely on hummingbirds and bats, which are unlikely to get stuck, unlike, you know, these bees and butterflies, which are easily stuck on sticky plants. We've also looked at stickiness across substrate and across sort of climatic factors that we have hypotheses about. we have not actually gotten particularly convincing data on this for a number of reasons. Our field data doesn't necessarily match up with our greenhouse data on stickiness, as well as our sort of quantification of herbarium specimens. And then the big project, and what I would say is my sort of white whale project of of my postdoc, is really gathering accurate data across clades and across the plant phylogeny, both for plant stickiness and insect entrapment, but also for seed mucilage. And looking at the literature, there's a few mentions of sticky plants, and I thought that this was going to be something I could easily do in my 2 years at Michigan State. And it definitely was not. And so I've gathered now thousands and thousands of species that have, but I'm not actually sure I'm that close to finishing either of these projects. And so the takeaways of this talk: stickiness is a really common trait. It's awesome. I love working on it. It's really fascinating. It's something that, when you show someone and get them to feel a sticky plant, a lot of people are really blown away by that. It's also really easy to work with. Basically, the first half of this talk, I used forceps and colored sand. And then, going forward, herbaria, as well as botanic gardens and seed collections, are really allowing us to take this ecology and ask evolutionary questions. And so thanks to everyone, especially Marge Weber at MSU, my postdoc advisor, and Rick Carbon at Davis, my PhD advisor. And I'm happy to take any questions now.
Great. Thank you for that really interesting talk. So just as a reminder for the audience, go ahead and put your questions in the Q&A. It looks like one of our ECSS committee members has a question. Brad, do you want to?
Sure, Eric. I don't necessarily have a question. That's really cool stuff, and I think about it. I just wanted to plug my particular group of interest. I don't know if you know of the river weeds or Podostomacea. They have sticky seed coats that allow them to distribute their seeds and get attached to rocks and rivers. And there's one species in the US that might be easy for you to get a hold of. But if you ever want to talk about in the future, or not now, but I just, that it's a really cool thing. And I'm glad you're working on it.
I would be really, so there's this this thing in the mucilaginous plant literature that it's a desert environment adaptation, and it's only useful in arid environments for water retention and all this other stuff. And it's evolved independently in your group. It's also evolved in some African wetland asters and a few other groups of wetland plants, which to me says there's something cool going on there. And yeah, I would love to talk about that. I'm really interested in its role in aquatic systems.
Anytime you want to talk, just let me know.
Okay.
All right.
I can't actually.
Oh, sorry.
I can't see the chat window for some reason.
Oh, that's okay. I will make sure that we get all the questions read. So we do have a question, a nice, simple, interesting one, which is, what is your favorite sticky plant?
Well, that's a really good question. I like the columbines a lot. They grow in these really wonderful serpentine seeps in Northern California, and they grow in the midsummer, and you're surrounded by all these beautiful California endemic species, and they're hummingbird pollinated. So there are hummingbirds buzzing around you, the whole time you're counting these thousands of dead insects. And it was just the best. It's the best plant to study that I've ever worked with, at least.
Cool. One question that I was thinking about is that I guess this kind of depends on the specimen preparation method, but with animal specimens that I've helped prepared, there's often a certain amount of cleaning that goes on. And I was wondering if you have to deal with that with herbarium specimens, did people in the past try to get all that sand off of the sticky plants? And was that a challenge?
Yeah, it's something I've really wondered about. I feel like it's something where if it's present, you can conclude that it's present, but if it's absent, you can't necessarily conclude absence of the trait. And yeah, I guess that's the thing that I've been operating on. I do feel like a lot of botanists, especially early botanists, were just wanted to collect a ton of specimens. And so I feel like the prep was lower in a lot of cases, and you find, like, I find enough dead insects and sand on specimens that I'm sure some people cleaned it, but I'm not sure it's super common.
Cool. Okay, another audience question. Is there any kind of arms race, quote unquote, happening between sticky seeds and insects that predate them?
Yeah, that's something I have no idea about. It's a great question. We've worked with harvester ants because they're really easy to work with, but we haven't actually necessarily worked with, well, some of the species we worked with were collections that I made and were native to the same areas as the harvester ants. But necessarily a lot of what we've worked with have just been commercially available seeds. So I don't really know if I think I'm capturing like ecological function, but I'm not sure if I'm capturing like, yeah, like actual dynamics between them.
Cool. Okay, another question, and maybe this will be the last one, or maybe one or two more quick ones if people have them. But so for this one, someone says, Louisa says, I work on parasitic plants, including mistletoes, which have very sticky seeds. However, some clades have evolved wind dispersed seeds. Have you noticed these cases of reversion in your work?
Yeah, there's a bunch of cases of reversion. So yeah, mistletoes have mucilaginous seeds that seem to probably help them in a number of ways, including like making it through the gut passage successfully. And yeah, there are a bunch of losses. Even if you just look in one genus like Salvia, there's a bunch of losses. I don't necessarily know what's going on there, but it seems like island taxa lose them, which kind of goes with what we know about, dispersal on islands, where a whole bunch of dispersal related traits are lost. Yeah, it'd be interesting. I have not looked at switches from, say, mucilage, which is a dispersive trait in some cases, but not always, to, say, wind or to water dispersal or to some other mode necessarily. But that would be an interesting thing once I get a much more complete list of these to think about. Thanks.
Cool. Okay. There are two other questions, but I think that we will maybe save those for the panel. Or Eric, if you can see the Q&A, you can also type out an answer. But for now, I think we should move on to the next talk. So our next talk will be by Laurel Yohe. Laurel Yohe is an NSF postdoctoral fellow in biology in the Department of Geology and Geophysics at Yale University, working with Anjan Buller. She is currently investigating the developmental basis of olfactory diversity in birds, reptiles, and mammals. She completed her PhD in evolutionary biology with Liliana Davalos at Stony Brook University, investigating the molecular evolution of chemosensation in bats. She has also spent a great deal of time doing fieldwork, including many expeditions to Latin America and to Vietnam as a US Fulbright Fellow. All right, I'll hand it over to you.
All right, thank you so much for the introduction. Can you see my screen?
Yeah, it looks great.
Okay, great. All right, so good afternoon, everyone, and thanks so much for coming to my talk. And thank you to the University of Michigan EEB for the invitation and allowing me to be part of this amazing symposium with some other really amazing early career scientists. And by now, I hope many of you have been able to see that from previous talks, the treasure trove of information that museum specimens can offer to science. So from locality information to parasites, there's the fantastic set of scientists within the symposium have set the stage for the value of just a single specimen. And I like to think about the story of the specimen. So for example, when this vampire bat was collected in 1953, it was likely intended for a few straightforward purposes. So perhaps for anatomy and species description, or scoring of morphological characters for systematics, documentation of locality, which can now be expanded upon for even some GIS modeling, and the parasite load, as we've seen in some of these talks, stomach contents for dietary information, and maybe even some ethanol preserved tissue that we can now use for genetics. And that's great. And we've expanded on that in every level we can think of, especially with the omics era. But the message is that the value of that specimen's worth may not even be recognized for years to come. And so when we collect the specimen, we need to have it preserved in every optimal way that we can possibly conceive. So for my talk today, I'm going to share both how new methods can inform new ways to preserve specimens, such as the onset of a lot of different omics techniques, genomics, transcriptomics, metabolomics, and how we can ensure that museum specimens are ready for this. But also how new methods can be used for old specimens, enlightening new disciplines such as Evo-Devo, which is what my postdoc is in new and innovative ways, or how specimens and collections may be used when it's all hands on deck, such as the case is what has happened in the pandemic. Now, I am a bat biologist, and a major part of my research investigates the molecular and morphological basis of olfaction in bats. And I'll go deeper into this a little bit later in the talk, but I first wanted to talk about how optimizing some of these techniques that we collect in the field that can be deposited into museums can sort of pave the way for new techniques that I've used in a lot of my work. And so focusing first on this sort of omics era, much of my previous work has relied on either freshly collected tissue from the field that then can get subsequently deposited into museums, or from field collections from the museums themselves. And I've been working with some of those curators such that we can now collect the tissues freshly in the ways that I might need for some of the projects that I've been a part of or have led. And what this actually looks like in the field is a lot more challenging logistically than is necessarily just spoken about. So this involves going into the forest, sometimes into very remote localities. So here I am with our field team in the cloud forests of Colombia. And we are working and catching the bats in the middle of the night, holding the bats in our hand, and then working to dissect right there on this plastic patio furniture of our hotel in order to optimally get out fresh tissue. And this is not just taking a liver sample and then discarding the rest of the specimen. This is taking the specimen, dissecting every possible organ we possibly can think would be of use, whether it's useful to my project or not, and then storing it in the case for transcriptomics in RNA later, and then flash freezing it in a liquid nitrogen tank that is just a lot of fun to bring home on an airline into and deal with customs and permits and etc. So it's a very extensive process, and there's a lot of different things that can go wrong. And so we want to make sure that every step of the way is optimized in the best way that we can. And I started thinking about how this could be streamlined when I started working with Bat1K. So Bat1K is a genomics consortium in which the initiative is to sequence every bat genome. So bats are special for a lot of reasons, and there's a lot of reasons to justify why sequencing the genome of over 1,000 bat species is worthwhile. But in order to take on this very large objective, it requires really freshly and pristinely preserved tissue. And sometimes, and the ethanol preserved tissues that was originally preserved in a lot of museum specimens isn't going to cut it for a lot of the new methodologies and high throughput sequencing techniques that need to be used for high quality chromosome assemblies. So I put together this protocol using the Journal of Visualized Experiments. They did a lot of the filming and production of this, where we actually developed a standardized method of dissecting out every organ throughout the entire body of the specimen and how to preserve it for different omics techniques. And this includes, for example, dissecting the brain. So instead of just taking the brain and smushing it in a little tube so that it gets pristinely preserved so it can be properly sectioned later on, so that actually regional parts of the brain could be analyzed, etc., as well as protocols for cell culture. So this is sort of the extended specimen, so life beyond the specimen, where we can actually have live cell cultures taken in the field and then brought back into the lab. And so for years to come, we can do expression assays or any sort of functional genomics using the tissue that was collected in the middle of the Amazonian cloud forest. So I encourage anybody watching to check this out, and if they can't get access to this paper or the video protocol, to contact me. But this is a message that is really important to me, to cherish every ounce of the specimen, to optimize for the future research beyond just the project that you're working on. So beyond just taking a liver sample and sort of, not really optimizing all the fresh tissue that can be obtained from that. So shifting gears a little bit to how I actually use a lot of museum specimens for some of my research questions. I'm referring to what I'm calling morphology 2.0. So sort of extending some of the morphology techniques that have been used in the past into new and innovative imaging techniques that we can do more so with in the future, and particularly focusing on embryology. So now to introduce you to my study system a little bit, I'm really interested in bats in the sense of smell. And this is not usually the first sense that people think of when they think of bats. So most bats are insectivores, and most bats rely on echolocation to find their food, such as this bat here that has landed this tasty moth. But there are several clades of bats that have evolved a different palate. So for example, within phyllostomins, the New World leaf-nosed bats, these bats are well known for their diversity of different diets. They're distributed throughout the neotropics from Mexico down through Argentina. And they live in all different types of habitats, from the desert to the deep into the Amazonian rainforest to up through the high altitudes in the Andes. And these bats are well known for their diversity in different diets. So there are bats that feed on insects within this clade, but there's also species that are more carnivorous, in which they feed on small vertebrates such as frogs. This includes the 3 species of the vampire bats, nectar feeding bats, bats that feed on a whole suite of frugivores, some of them only feeding on soft fruits such as piper, or others that are more specialized on hard fruits such as figs. And to accompany these consumption of a variety of different dietary resources as a suite of morphological traits to complement the consumption of this food. So we have bats with strong bite forces to bite through figs, or we have nectar feeding bats with their long palates and sticky tongues. But it's not really well understood exactly how all these bats find their food. So echolocation is really useful for finding a flying insect in the open air, but it's less useful for finding a fruit that might be a stagnant fruit that is hiding under a leaf in a cluttered forest in the dark. So we know that these bats must be using something else other than echolocation. And behaviorally, we do know that these bats use primarily olfaction to search and explore the environment. And when they locate their food resource, they actually supplement with echolocation just to orient themselves to land on the fruit or grab the fruit and then fly away. So olfaction is a very important sense to these bats that feed on plant compounds. And so this has inspired me to try to understand how the sense of smell might differ in bats that feed on plants versus those that feed on animals. And that includes insects or some of the carnivorous bats as well. And just a brief introduction as to how the sense of smell works in most mammals. So whatever odorant cue the animal breathes in, it enters the nasal cavity and it deposits the odorants onto the main olfactory epithelium that are distributed along these turbinates. And these turbinates and the olfactory epithelium express the olfactory receptors, which I've done a lot of work on as well. And that was a lot of the genomics work that I had collected tissue for that I don't have time to talk about today. But I'm also interested in the morphology of these turbinate bones where the olfactory receptors get expressed. And so to visualize these, I wanted to come up with a way to visualize these very delicate bones inside the head, but not just the bones, also the epithelial tissue, the olfactory epithelial tissue that is distributed on top of it. And at the time when I was interested in this, I was at Stony Brook working with people in the anatomical sciences department, and they were developing this technique called DICE-CT. So this iodine staining contrast CT scanning. And what this means is, if we're looking at, for example, this alligator head, if you were to put this specimen into the CT scanner without any sort of treatment, what you would mostly be able to resolve is the bony tissue. However, if you soak the specimen in iodine or any other sort of contrast stain that is available, what you get is incredible resolution of the soft tissue. This includes musculature, this is the brain here, you get blood vessels, neurons, and a whole suite of morphological traits. And so if we look at this in sagittal view, you can see we get amazing resolution of the juvenile crocodile brain. You can see all the neurons inside the tongue. Every time I see these, I can spend hours looking at these CT scans because there's just a wealth of data. And it's just has become available to us using a museum specimen that has been sitting there on the shelf, but it just is being presented in a new and exciting way. And so I wanted to see if we could then visualize these olfactory turbinates of bones, or these olfactory turbinate bones on a distributed epithelial tissue. And indeed, what I have been able to describe is that you can definitely see these bones using this contrast CT scanning methods. So this is in sagittal view, and these different bones, or these different colors correspond to the different turbinal bones. And you can see the squishy epithelial tissue distributed on top of it. So in bats, there are usually, or in phyllostomid bats especially, there are about 5 turbinal bones. And then, and that's what is color coordinated here. And the olfactory epithelium is then subsequently distributed on that. So what I do with the CT scan is each individual section, I trace around what I think is the epithelial tissue. And then when you put it all together in this 3D stack, you get this really amazing 3D structure of these very complex morphological, a very complex morphological trait, very difficult to study without using this method of these very delicate bones. Whoops. Let me just So the question that I was asking is whether or not the animal feeding bats had smaller or lesser developed turbinates compared to plant visiting bats. And this project involves combining both genomic data with the olfactory receptors, which I don't have time to explain today. But I have a complementary data set of the genomics and the morphological data set for about 31 different species of bats with different diets. And just to show some initial results, we're about to submit this paper here. And what I want to focus on is the results is that we do indeed find, after controlling for body mass, after controlling for phylogeny, after controlling for measurement error, after controlling for the variation of the olfactory receptor molecular evolution, we get this amazing relationship of these hypervariable turbinal bones. We actually do indeed see a difference in the animal and plant visiting bats, such that the olfactory surface area epithelium is greater in plant visiting bats than in animal feeding bats. And if we look at, if we just look at this qualitatively, it's actually quite striking, the difference in both the size and the shape of those 5 different bones. And so I wanted to be able to study this in a more detail. This is to me, there's a lot more questions to be asked, particularly within the morphogenesis of some of these olfactory turbinate bones. So if we look at these individually, this is phylogenetically corrected ANOVAs, and after controlling for multiple comparisons, what you get demonstrating the surface areas of these each individual turbinate bone or epithelial surface areas on these different turbinal bones is that the blue and the green and the purple tend to not differ so much in animal and plant visiting bats. However, these orange and these pink seem to be the most variable across the 2 species. And so to take a deeper look at this, I'm going to turn to a very undervalued research in museum collections, especially in mammalian collections. And that is of females. So one additional resource that a female specimen offers is potentially more than one specimen. And collecting embryos can be very costly to bat populations, especially given that bats only have 1 to 2 pups per litter. There's extensive maternal investment, which is unusual given their body size. So thus removing a mother and an embryo could have significant long-term impacts. And this is where museum specimens have become extremely valuable. So it is possible through minimal invasion of a small incision of a female specimen that you can discover an embryo within that female. And that has been sitting on the shelf for decades and we don't even know it. And in fact, this is precisely what Dan Urban, who works at UIC Urbana-Champaign, did. So I'm very grateful to him. He sifted through hundreds of female bat specimens, identified hundreds of embryos, staged them, and they're now accessioned in the American Museum of Natural History. So I've taken a subset of those, and about 100 or so, and for about 10 or so different species, and I have about 6 to 8 embryos at different stages. And I wanted to see how the iodine staining and contrast CT scanning work with these tiny embryos. And lo and behold, what you get is this incredible resolution of these tiny bat embryos. And we can look back, in developmental time, and we get a brilliant window into the internal morphology of a developing bat, even into the earliest stages where the brain is not even yet present. I find these absolutely breathtaking, and there's so much to learn from them. So just focusing on the project at hand here, though, we can indeed see these turbinate bones developing in the embryo. And if I look at this across species, I'm just going to present some preliminary results on this. If we look at bats that feed on hard fruit compared to similar ones that are feeding on nectar and ones that are feeding on insects, an important pattern I think is emerging. And for one, the purple, the green, and the turquoise turbinal bones look relatively similar across the 3 species. What tends to be lesser developed, even later in the embryonic stages, are those pink and those orange turbinal bones. So I think I might be onto something here, in which these hypervariable ones that are showing the most difference between the plant visiting and the animal feeding bats are actually occurring on the turbinates that are developing much later. And this is a characteristic that is observed throughout Evo-Devo. It's a trend that is observed throughout evolution and development, such that later developed phenotypes tend to be more variable or more subject to selection. So I think this warrants a deeper look, and I'm really excited to look at the entire series across the different specimens. So in summary, female specimens offer like, you know, unlimited potential for embryo series, and I think this is something that needs to be expanded upon in the future. Okay, so for the last part of my talk, I'm going to spend some time on a part of a talk that would not have existed had we been able to hold the symposium last year. And it was pretty much exactly at this time I was going through this thought process. So not more than a year ago, we were all glued to our screens, waiting to see what the fate of the world was going to be. And as the weeks went by, for me, there were two patterns that were emerging. First, bats were being described as the emerging reservoir for the coronavirus. And two, everyone was losing their sense of smell. So suddenly being an expert in bat noses was becoming increasingly relevant. So it turns out that bat noses actually have much more in common with human noses than our current rodent models. And this inspired a team of us to put together an NSF Rapid grant to investigate why bat noses were, or why bats were able to become infected with the coronaviruses and not manifest any symptoms, while humans are infected with the coronavirus, and then, you know, everyone knows what is happening there. So I like to acknowledge just this powerhouse team of us that are looking at this, and it's been a really exciting journey to see how the data that we already had available has been applicable to this project. So like I said, bats are actually a great model for understanding the human nose for two major reasons. So first, humans and bats have a continuous nasal chamber in which the air is inhaled through the nose and makes streamlined contact with the respiratory and olfactory epithelium, where this is not the case in rodents. In fact, the respiratory and the olfactory epithelium in the rodent model is actually separated by a bone. So we can imagine how this airflow transfer of the virus would differ in the cells within the nasal passage. And second, there's a special organ in the nose called the vomeronasal organ, which is highlighted in pink here. And it's present in most mammals, and it's mostly used in social chemical signaling, and it's included in rodents, but it's actually lost in most bats, though there are some bats that still retain it. So it makes, this variation makes for some intriguing comparisons. But this loss of the vomeronasal organ and this continuous nasal passage makes the model of the nasal system much more comparable and familiar to us in terms of trying to understand how the virus is invading. But perhaps what is the most important about these tissues within the nose is that they're jam-packed with mucus-producing cells. So enter the goblet cell. Goblet cells are these often overlooked cells that may be key players or in the coronavirus infection, as they're sort of ground zero for viral entry. So goblet cells are long thought to be mucus-producing bystanders in disease pathogenesis, but they're actually probably much more important than is previously thought. So first of all, they express high amounts of ACE2 receptors, which is the receptor for viral entry. And there's a differential severity of this expression. This difference in this expression can be related to the differential severity of the COVID infections. So turning to our scan data, we can actually see the distribution of where goblet cells are concentrated. And these CT scans will inform multiple parts of the project. And the way that we have the project structured, we have several creative approaches for investigating goblet cell function across different species of bats, the rodent models, and the humans. But one of the problems with our initial data was that well, it's not necessarily a problem, but one of the limitations was most of my samples were neotropical. And in order to sort of get a proper comparison, especially of bats that were the presumed reservoirs of the coronaviruses, we needed access to Rhinolophus or horseshoe bat samples of these old world species. And so this is where we turn to the museum. And I have to be eternally grateful to especially the Museum of Natural History in New York. They were like sort of first responders in terms of trying to make sure these high demand specimens were delivered as soon as possible. So we were able to get a really nice handful of Rhinolophus or horseshoe bat species to do a proper comparison across the different bat species, but also within the bats of humans. And what was so amazing to me was when we first received the specimens, is actually how old they are. So for example, the one that I'm going to show in just a second here was sampled in 1916. And so I was thinking, well, is that going to have any sort of effect on any of the methods, these like pioneering methods that I had proposed to do? And so I thought, well, let's just give it a shot and see what happens. So first, just doing the iodine CT scanning of this Rhinolophus bat. We actually get this is a sagittal section here, incredible resolution of the nasal cavity up here. So here are the teeth coming in. This is the brain. Here's its amazing cochlea. Here's the tongue. And you can see these guys are sort of like superstar echolocators. You can see just how much the cochlea takes up of their head. It's pretty remarkable. But I can safely say that the iodine CT scanning has been extremely valuable, just given especially the age of the specimen. It has seemed to have very little effect, and particularly on the nasal cavity tissues. Even just a short staining session, we're getting absolutely brilliant resolution of, so these glowing right here, this is part of the respiratory epithelium, and these it's just, and compared to what we see in the olfactory tissue back here, just the differences of how these tissues are distributed in space, I think are going to be very informative to the immunohistochemistry to come. And I just want to point out the incredible resolution of the skin cells and the hypersensitivity within these bats is just incredible and warrant a lot of future investigation. So the second part of the especially comparative anatomy was to do some histology of this. And so again, like these are pretty old specimens and I wasn't sure what was going to happen. And also I had like no idea how they were preserved, whether they were in ethanol, whether they were in, they were in formalin, there was no real documentation exactly how these are fixed. So taking this sort of pickled bat and really having no clue of the history of its preparation to the pathology department here at Yale, I got a big fat no that this was not going to work. But I insisted and said, I'm paying for it, so just give it a shot. And lo and behold, what we get is, this is just an H&E section of a histology stains. So here's, and this is just the lining of the nasal cavity. So before I point out some of the interesting features relevant to the project, I just want to point out this like amazing blood vessel here. And I mean, these blood cells look like they were sampled just yesterday, but in fact, this bat was pickled in 1916. So this is over 100 years old. But focusing on this epithelial tissue here, we get beautiful resolution of the cilia, and we're teaming with people at John Jay, and we're using some deep learning and some exciting algorithms for identifying the goblet cells within these structures. And then the next step then is to do the immunohistochemistry. And so I think so far, by just giving things a shot and not knowing what you're going to find, it's been very rewarding. And so I just just to wrap up here, museums are a powerhouse of resources when there's a sudden shortage of material. And I'm very grateful to the people who have made this possible. But I also want the message to be like, you don't really know what your limitations are unless you try. People are always going to try to stop you, but you know, you never know what the real case is going to be unless you try it for yourself. So reflecting on back with this vampire bat specimen, and what is the value of when we sacrifice for the sake of specimen collecting. What I've learned is that we are not limited by new methods if we prepare and preserve properly. And we are not limited by size, and we are not limited by age. So while things might seem like they're phasing out, we're only just beginning. And it just is a matter of innovation and application and creativity to see what we can discover. And on a final note, I want to emphasize how we're not limited by ideas either. So all of this pandemic related work never would have come about unless I had been studying bat noses, which I had only been studying because I thought were really cool and interesting, and I love sensory biology. So it's not because I had any sort of underlying translatable agenda for human benefit. And I want my message to be that investing in basic research just for the advancement of knowledge is always worth it, as you never know what you're going to discover. And it just may turn out that bat noses can save our world. So I would like to thank all of my funding sources, especially the NSF. I had an NSF Specimens postdoc fellowship, so I'm very grateful for that, to explore all these questions, as well as a lot of my different collaborators, and particularly those in the museums that have helped make this possible. And thank you for your attention.
Great, thank you. That was a really cool talk. So we've got a couple questions already. Again, you can, if you have a question specifically for the first 5 minutes or so of this next part of the symposium, we'll focus on talks, questions about this talk in particular, and you can put them in the Q&A. So the first question we have, it says, I presume that you are keeping voucher specimens associated with all the dissections and tissues you are collecting, and associated with that, are you also collecting external parasites? And then I guess there's a second part to the question, but I'll ask it when you're done.
So yes, usually yes. So usually on these collecting expeditions, we have permits to collect 2 to 3 individuals, usually males. And what we do, and this is detailed in the publication, is to have a voucher specimen, and oftentimes that is usually the one that I use for CT scanning, or like in the case in our expedition where we went to Peru, we had a voucher specimen that was deposited into the museum there. They received parts of the tissue that we exported as well, of a different specimen. So there's sort of a hierarchy of replication of the dissections that occur. And usually, if it's a rare specimen, we might not even have the permits to collect it in general. So usually the permits that are usually we have 2 to 3 replicates per species that are used for these different protocols. But we really try to keep up with the different protocols, even if I'm not doing metabolomics, understanding what the procedure would be to preserve that different tissue for someone who might want to do metabolomics, for example.
Cool. Yeah, I think that partially answers the second part of the question, which was asking about balancing, you know, the CT scanning of the whole animal versus dissecting out some tissues.
Yes, no, but I agree. Like you definitely can't do both, and it is destructive of the skull, etc., to do the full dissections, especially if you want to have the brain. But it's the trade-off of, do we want to not have any sort of brain for this species in order to do any of sort of the neurogenomics that is now available, spatial transcriptomics, etc. So
cool. Okay. Another question is, this is fascinating. I love your methodology, in particular the way you describe cherishing every ounce of the specimen. When it comes to conservation in particular with threatened or endangered species, what guidelines do you follow in determining how many specimens to collect and whether specimens should be collected?
Yeah, so that's a really good question, and it's kind of related to the last point. First of all, it depends on where we are doing our expeditions and what we can get permits for. And usually any sort of endangered species is like kind of off the list, unless there's a strong case for one way or the other. But when we write our permits, we usually ask to do 2 to 3 specimens per species, and usually males, to avoid sacrificing a female that might potentially be pregnant. And so that, again, that's sort of the hierarchy of how we think about that. But every time we do sacrifice for specimen deposit, I like to make sure that like it is going to be maximized. All of its value is going to be maximized in every possible way that we can think of at the moment.
Great. Okay, actually, so we have 2 questions now, both with kind of the same theme, which is about the iodine associated with the DICE-CT scan. So I guess to sort of synthesize these 2 questions, the first is, does the iodine stain harm the specimens?
Yeah, so I forgot to mention that, I can't believe it, but one of the major advantages of especially using iodine is that it's diffusion based. And so when you're staining it, the iodine will bind to different tissues within the specimen at different contrasts, or at different concentrations. And then that concentration of iodine will reflect or show different contrasts with the X-rays. And so while the iodine, it's dissolved within potassium iodide, so it's actually just disassociated, and so it's easily diffused in and out of the tissue. So when you first put it in stain, the specimen kind of sucks it all up, and then depending on how much you stain it, or if you refresh the stain, more iodine will go into the specimen. But you can unstain it. It is diffusion. So putting it back, once you're done scanning it, it is possible to unstain it. And there's different ways you can use different salts to further extract it. And it will eventually unstain. It's just a matter of patience. If in terms of damaging the specimen, it is possible that that there is shrinkage involved. And a lot of this has to do with the differences in how the specimen was preserved. So if you had it in formalin and then put it in ethanol, or if you had it in ethanol and then it was in methanol, and those sorts of things tend to affect how much shrinkage, mostly of the brain and neural tissue. Like you can see here in this bat behind me, like the brain is sort of separated a little bit from the skull. That's due to shrinkage. And so that can technically damage the specimen, but a lot of that has to do with the fixative more so than it does with the iodine.
Great, and that answered the second part of those questions as well, which was about whether or not it can be removed once it's been stained. So I have another question from one of our ECSS committee members, which is, as it relates to coronaviruses, there are other bats that are hosts, other than the ones that you were talking about, I guess. Have you compared those taxa, such as philostomids, with your rhinolophid data?
Yeah, so actually, we have this beautiful histology data set, also museum loan, from a previous study of phylostomid noses. And so that was sort of our starting point in terms of understanding where goblet cells were distributed in bat noses. And it was us preparing the rhinolophid noses to compare to those is sort of what has been the most recent thing that we've done. So we have about 10 species of phylostomids in histology, and then we also have all of the CT scans. So combining that all together, we actually have a really nice data set across many different bat species. One general reflection that we've found is just how incredibly variable the bat noses are in terms of tissue types, tissue distribution. I mean, it's just the types of epithelial tissue within the nose are extremely underappreciated. And even within the Rhinolophus species, I think we have like 3 Rhinolophus species that we've looked at so far. And just across those 3 species, you know, a lot of times you kind of dismiss things like Rhinolophus or Myotis as being like, oh, very similar phenotypically. But actually, when you look inside the nose, it's vastly different. And so just sort of sorting through the functional relevance of that variation and what it can mean, I think is interesting in and of itself, independent of the coronaviruses. But I think there's going to be a lot to be learned in this comparative approach.
Cool. Okay. One thing I guess that I thought was interesting about your talk was how you were able to use the embryos that maybe people didn't even realize were in those specimens. And you mentioned also that female bats are collected less often. So it's always interesting to think about like what biases in a lot of vertebrates. I think that males are typically targeted, whether for like conservation reasons, like you've mentioned, or in birds, which I study, they're often more colorful and people are kind of more interested in those plumages. So I guess my question is, as you've been thinking about like studying development with these CT scans, has that changed your sort of philosophy about like what to target when you're doing collecting for museums?
Mm-hmm. Yeah, so that's a good question, and it's a delicate topic for sure, because it is something that I and many bat biologists and conservation biologists are aware of the implications for what it can do to a population of bats potentially. I think what is merited is starting with the museum collections. There are like, I just find it amazing that you just don't even know what like how many embryo specimens are potentially sitting there. So I think in order to justify collecting deliberately for embryonic and ontogenetic series of bats, I mean, of course there's so much to be learned, and in vivo in and of itself is an emerging field, and it's just starting to get its head on straight in terms of like proper quantitative analyses, methods, and finally actually obtaining series of embryos and staging them and phenotyping them. And so I think starting with what is available in the museum and seeing what is possible is more justifiable right now, especially like in the pandemic where no one is able to do field research right now. This has been really like a useful way to use my time. So I think it can inform where gaps are. So if for example, in a collection, you don't have a lot of, within one species, you weren't able to find any embryo series of this, then of a species where you can potentially get multiple replicates of, I do think it could inform collecting trips better when you have a sense of what is available and what you can do with what is available. That being said, I know from several collaborators that we have who have done embryo collecting, it can be really challenging to determine when is most appropriate to collect the females, because you do have to know when they will be pregnant. And then in order, if you were to want to have a developmental series as well. So I think collecting in a place where you've already been collecting, where you're familiar with the taxa of what you're going to collect, and potentially the breeding seasons, would help inform responsible collecting of that as well.
Great, yeah, that makes total sense. All right, I think now we can transition to the more panel focused aspect of today. So Eric, you can come back whenever you're ready. And those of you in the audience are still welcome to ask whatever questions you have in the Q&A. And especially if you have questions that maybe are bigger in picture or focused on early career issues or issues with museum collections in general. And I guess I'll start off with a question that both of you can answer, which is both of you are using museum specimens in, I think, really interesting and novel ways. And I was wondering if you could reflect on your development as a scientist and what kind of like opportunities led you to have these novel uses of museums, like where those opportunities came from and where those ideas came from.
I can start. So for me, I guess if there's students watching, this is particularly relevant to them. When I was an undergraduate, I was a bioinformatics major. And so I actually joined a phylogenetics lab for like the interest of doing bioinformatics related phylogenetic inference. And so I had no idea really anything of the scope of a field collection or species description or what a museum had to offer. And actually my undergraduate mentor, Sushma Reddy at Loyola Chicago at the time, she worked at the field museum. And one day she just like let me tag along to see the specimens. And I didn't even know stuff happened behind the scenes at museums at the time. So it was absolutely mind blowing. And I just fell in love with it since then. And so being exposed via a mentor who actively field collected and also her own samples and also worked in the museum environment, that was very inspirational for me. And then my PhD advisor, Liana Davlos, who works with bats, I tagged along on those field expeditions and, you know, working closely with her advisor was Nancy Simmons at the Museum of Natural History. So just really having strong mentorship early on and realizing the potential of what is available has been really important to me since then.
Yeah, this I guess is something that I come at this from a very like field ecology perspective, and I think the collections are very much underutilized. And I don't think if I hadn't like, I guess I also have an entomological background, and I would say that like ecological entomologists are much more collections oriented than plant ecologists. That's not necessarily to mean that there aren't a lot of plant ecologists that do contribute to and use herbarium collections. But I do think it's a little bit better integrated in the entomological world. And yeah, I guess I really came at it. I mean, I've used herbaria to identify stuff for a long time, and it just seemed like the sort of natural way to start asking these evolutionary questions when I decided that, hey, look, I have one skill, field ecology, and what do I want to do for a postdoc? You know, and I did contribute to the herbarium because, you know, as someone with a background in entomology, it's like, well, you take voucher specimens and all. Yeah. I guess that's sort of my sense. I think it would be, I don't know how to get collections more integrated into field ecology, but I think it would be a good thing to do. And I think that there's a lot of sort of stuff, like really awesome information locked away that could be really useful.
Yeah, it's interesting that I guess you definitely come at it from a field that, as you were saying, tends to have fewer people maybe intersecting with museums. So it is interesting to think about why you're able to do that. And I guess it sounds like it was through the entomological lens. But yeah, do either of you want to expand more on like what people associated with museum collections now can do to make sure that the collections are being utilized to their full potential, and also I think appreciated? Sometimes that's something that collections struggle with. I mean, they're these amazing data sources that are also like often kind of being downsized by universities, or you know, I think that's a challenge that a lot of collections have faced.
Yeah, that's a good question. I think spotlighting different ways that they are being used in like, I think especially what is sort of expanding in sort of a dramatic way is this iodine staining with the CT specimens, because CT scanning has been a huge part of museum specimen research ranging from fossils to just skulls, anything that is kept in there, and first of all, for digitizing reasons, but also for just whatever morphometrics and cranial evolution and mammals, for example. Every CT scanning was huge and still is. But now with the iodine staining, there's so much more anatomy to be looked at that has never even been fathomed to be considered. And so pretty much every museum that I've worked with in terms of trying to request a specimen for the iodine CT scanning, although it is just technically destructive sampling, they've been very enthusiastic about using that approach. And I think that's a big avenue that has a lot of application. And one thing, for example, that has been really cool for me with the coronavirus thing is using the scanning methods. I've been exposed to a lot of other scientists, and actually the pathology people at Yale were like, wow, that's super awesome. Like, how are you doing that? And so actually I'm using that technique on their hamster specimens that they have with the COVID infections. And we're actually looking at the neuropathogenesis. You could see the neurons using the CT scans. So you can see if there are neurons that are inflamed. So, you know, it potentially has clinical implications using techniques like this. And museums can provide those resources. And so I think the sort of internal anatomy, I really am hoping that anatomy in and of itself, which is also for a while was, especially like Stony Brook, it was a big drama of like the decline of anatomical sciences. And now is the time to be like, anatomy is important here, okay? Looking at a rodent head is not useful when we're looking at trying to understand how the coronavirus gets inside the brain. Like, there's just not comparable. Like, it's not showing us what we want to see. Like, maybe we should look at a different specimen, or a different species where this might be more relevant. And that is where I think museums can play a huge role.
Yeah, I guess in terms of like demonstrating the value, I really don't think it's like my sort of low technology, just looking at sheets kind of research. I think it is really, integrating, whether it's genetic stuff or this, all this really cool methods of visualization. And then the like push for extended specimens and getting, all of these different data sources integrated. I think that's the way that's going to get new people excited about collections, because of the sort of big data and the amount of sort of integrative data that can come out of it.
Yeah, well, and I guess your work is also kind of intersects with the extended specimen concept, because you're looking at all those insects that are stuck to them. We have a question from the audience that's actually pretty along the same lines as what we've been just talking about, but it sounds like it's someone from a small collection. So they ask, do you have recommendations for small collections with limited funding to be sure our collections will be maximally useful for a variety of potential research in the future?
Yeah, I think that's a really good question that I'm sure a lot of small collections think about. And I would say one thing that is very valuable about small collections is they often have a lot of like one type of thing, or they tend to have lots and lots and lots of replicates of a small subset of species. So they might not necessarily have the extensive diversity that a larger museum might have to offer, but they tend to have a lot of replicates of different things. And as some of these methods are becoming increasingly relevant to the comparative anatomy, so I do a lot of embryo work with other staining methods outside of this, with confocal microscopy, and some of the histology and stuff that we were doing. And that requires, in order for publication, to have multiple replicates of something for. And so it's not always easy or possible to have multiple individuals of a same species requested for something. And so I think making that known of what you have a lot of is something that will become increasingly valuable as well. And just in connection to that, in terms of an evolutionary perspective, we're also at a time where the bridge between microevolution and macroevolutionary processes is becoming stronger and stronger. So connecting this population level phenotype genotype with a macroevolutionary comparative scale is definitely a hot spot of research. And where if you would have lots of replicates of one type of individual or one type of species, you know, I think that is definitely one way to shine.
My answer would be totally different on this one. I think that the beauty of these small collections is how they document the local flora and fauna and changes, and how, yeah, I don't know how exactly to get people to care about that, but that's why obviously they're such a good resource. And I don't know if, like, there was a really nice study a couple years ago that looked at, I think it was heavy metal contamination on plant specimens and showed sort of pollution levels over time. And like that sort of ability to look at the history of a given local area and stressing that to a university, museum, et cetera, I think would be, I don't know if it's a way to sell it, but I certainly see that as a major value of those smaller collections.
All right. Another question that's kind of centered around the stage of your career, I guess, is as you transition from the stage you're at now into, you know, potentially having a lab and your own students in the future, how do you envision continuing to use collections and engage students with them?
I mean, I think they should be used in teaching, and so I'll be teaching field botany here at Oklahoma State, and we have a teaching herbarium just for that. And I think it would be really nice if, you know, both plant and animal collections were more commonly used in classes, you know, across various parts of the university. There was at Texas Tech, there was the curator there is using collections to have students build phylogenies using this new sort of rapid sequencing technology that I cannot, for the life of me, remember the name of. And he's found that to be a really cool tool. And so I think encouraging that sort of use would be, you know, useful for continuity and introducing students who end up in sort of broad disciplines.
Yeah, for me, it's museum collections will continue to be indispensable for my work. And what has been really amazing for me is that the more I do of one project, it sort of opens up the avenues for the next project. And all of this happens just out of like the pursuit of curiosity. So I am a genomicist bioinformatician by training. I started the iodine CT scanning because there were people at Stony Brook doing it. And I was like, hey, I have a bat specimen that I just got in the Dominican Republic. Like, I want to see if this method works on my animal. And like, look where it's gotten me. Now I'm doing it on hundreds of embryos of museum collections. So I think for me, it's sort of, I don't exactly know what the next step is going to be, but I like knowing that resource is available and, you know, continuing to want to contribute it to it myself, and optimizing ways of preservation and as well is something that I intend as an early career scientist to incorporate. and that is not just with the anatomy, it's also with the genomics, and keeping up with the methods and how we need to preserve it, and helping to communicate that with the collections as well, is something that is important to me in my research vision.
Cool. I'm curious if either of you have ever interacted with high school students or younger students who are in college with museum collections, and if you have any ideas about how to inform younger students about like the existence of collections even, or the fact that we still use them, and what kinds of questions we can address with them.
Yeah, so actually Stony Brook has, where I did my PhD, has an amazing like hybrid high school research program where like the high schoolers are paired with graduate students on different projects, and they have these science fairs that are like way beyond a science fair that I ever presented at in grade school or high school. And so they're actually doing a minimum undergraduate level research. Sometimes they've been like graduate student level projects. And that has been, that was a really special experience for me. I've had 2 to 3 of them where they have been able to work on the CT scans. Something like this, the CT scan data, it's just, the things that one can look at within it is somewhat limitless. And I think you can have multiple students working on the same scans or the same specimens, but in different ways that can help inform one another. But what was also cool is that they kind of saw the processes happening from the beginning as well. So they were able to see the specimens, they were able to see the staining, and how all of that happened as well. And I think museums are a great start for introducing either whether they're high school students or undergraduates to research, just because you have such a wealth of data that has already somewhat been collected for you. You just need to extract what you need to for the project at hand. But I think it's a great resource for teaching people how to do research.
I don't have very much to add. This is not something I've done anything about, but I wonder if, like, I have worked with students on, this past year, I have worked with students on remote projects that, took like GBIF and iNaturalist data, and I wonder if there's some sort of integration, if you can assign students that and really stress that, these data are not just like points on a spreadsheet. They're, like a real world specimen that exists in, this herbarium somewhere. But yeah, that's about all I got for that.
Yeah, there is something to be said about like holding something in your hand versus doing something remotely. I mean, I think anybody who's done any field work or worked with museum collections can say that there's a difference between looking at a spreadsheet, exactly as Eric said, than actually seeing it.
Great. Well, this has been a really interesting conversation. I think we'll probably wrap it up soon. So if anyone in attendance has any last questions, please make sure to get those into the Q&A as soon as you can. And I guess while we wait just a few moments to see if there are any other questions, do either of you have ideas of crazy like new ways that people might use specimens that we have, that people aren't even doing yet?
I guess one thing that I've been doing in my lab at Yale, and it hasn't necessarily been yet with museum collections, is doing this type of protocol called clearing, and so you clear and stain it. Basically, if there's a particular particular structure like muscles, or if there's a particular protein you're interested in, you use the antibody to target whatever structure or potential protein you're interested in. And then you use different methods to prepare the fixed specimen in that way. And then you clear it, and basically the specimen becomes invisible. And then you image it on a confocal microscope, and you get brilliant resolution in 3D of that specific structure. So in contrast to what the DICE-CT scanning is, you get, I'm just gonna, if I still have screen sharing privileges, I'm just gonna show an image. I
think, yeah, there you
go. Okay, yeah, so this is a chicken embryo, like a late stage chicken embryo, and it's stained for PAX3 gene, which stains a part of the neural crest that's developing. And so you can see exactly where it's concentrated, and then you could actually sift through this in 3 dimensions and see exactly where in the brain this is. And personally, I don't see why you couldn't do this with a museum specimen. This is from a chicken embryo that we collected in the lab, but I mean, it's no different than going to the field and collecting it, and it's no different than like I tried with that specimen from the early 1900s. You know, you never know what's going to happen. And I think that applications of this clearing and imaging has a lot of potential.
Cool. Yeah, that was a really cool image. All right. Well, thank you so much for your talks. They were both really interesting, and thank you everyone for attending. And I guess we'll see you next week when we have our final talk of the ECSS, our last keynote by Pam Saltis.
Yeah, thanks for having me. This was fun.
Yep, thanks for the invitation. And I'm glad this event was able to take place after all.